Good afternoon everyone. Myself Mrs. Ashwini Kadbhane from the Department of Microbiology S.V.K.T College, Devlali Camp. I am going to teach you TYBSC paper number 3, that is the metabolism from semester number 6. In metabolism, today we are going to see the first unit of the metabolism, that is the membrane transport. In membrane transport, in today's session, we are going to learn about how the transport of various nutrients in the bacterial cell is going to happen. So, here we are going to see what is the cell membrane and how the transportation mechanism is done in the bacteria. Before starting the today's session, we have to see what we have learned earlier. In our previous lecture, we have already learned about the enzyme purification in enzymology. In the paper number 3 only, in your last semester you were having the enzymology subject. In that we have seen about the purification of enzyme on the basis of its size, its charge, its solubility differences, its specific binding property and the selective absorption. In our today's lecture, that is the next new semester, semester number 6, paper number 3, metabolism, we are going to start from our today's lecture. In today's lecture, we are going to first see the introduction about the plasma membrane, its structure, its components and the function of the plasma membrane. In plasma membrane, we are going to see about the fluid mosaic model, which was a standard model for the plasma membrane. We are going to see its characteristics and its function. We are going to see the membrane transport mechanism, various mechanism. In that, we are going to see about the active and passive. Today we are going to focus on the passive type of mechanism, which includes the simple and the facilitated type of the diffusion mechanism. Okay. In that we are going to elaborate that, and we are going also to see that the direction of the transport, which type of direction is there in this facilitated and the simple type of diffusion. And the last but not the least, we are going to see about the distinguishing characters of active and passive transport. So, now we are going to see our today's session. In that, we all know that every cell except the eukaryotic cell, every prokaryotic cell is having a cell wall and then the cell membrane. There is absence of the cell wall in the eukaryotes. Eukaryote doesn't have the cell wall, they directly have the cell membrane. But in prokaryotes, there is the cell wall, and after the cell wall, there is a cell membrane, which is the selective or semi-permeable, and which is also called as the plasma membrane. Plasma membrane is also a semi or the selective permeable membrane. Plasma membrane is also a cell membrane which has various functions, but the most basic function, that is to keep the cell functional. To keep the cell function means to transport the various nutrients from outside to inside and from inside to outside the cell. The main function is carried by the plasma membrane, which we are going to see in our today's session. The plasma membrane is selectively permeable. Selective permeable means only some things can be permitted to go inside the cell and to come outside the cell. Not every molecule or every substances can do this. So, it is called as the selective permeable, because only selective molecules have the permission to pass through the cell membrane. This means that membrane allow only some materials to freely enter or to leave the cell, while the other materials cannot freely but require use of specialized structure and occasionally even the energy investment for the crossing over. Now we are going to see the components and the function of the plasma membrane. Now the first component, the phospholipid. Before starting this, I am going to tell you that the plasma membrane is comprised by the proteins, lipids, carbohydrates, okay, glycoprotein and glycolipid. They are the two components which are present in the plasma membrane, which includes the carbohydrate plus lipid and carbohydrate plus protein. Also, it includes the phospholipid. Also, it includes the cholesterol, peripheral proteins and the various lipids. So, phospholipid, it is the major components which have the main fabric of the membrane. Cholesterol, it is attached between the phospholipids and between the phospholipid layer. Integral protein, for example, integrins, they are embedded in the embedded means they are in interior of this plasma membrane. Peripheral means which are on the surface or on the outer surface of the plasma membrane, but integral protein means which are inside the plasma membrane. So, integral protein which are embedded with the phospholipid layers may or may not be penetrate through both the layers. Peripheral proteins on the inner or the outer surface of the phospholipid bilayer and not embedded with the phospholipids. Carbohydrates, they are generally attached to outside the layers of the phospholipids. So, these are the major components of your plasma membrane and their various functions. Now, we are going to see about the various function of the plasma membrane. Now, all of you know that the major function of a plasma membrane or a cell membrane is to keep the cell separate from the environment, because it is trying to keep the cell very preserve and protect the cell from the outer tensions. That if the concentration is more outside, the cell permeability or the plasma membrane allows only the solute to transfer from outside to inside till the equilibrium is not attained. That is the homeostasis. Homeostasis is if achieved, then there would be no transportation or no diffusion of various molecules from outside to inside and inside to outside. So, this is the major function, that it separates the cell from the environment, it transports the substance from in to out and out to in, it receives and responds to the stimuli. Because when there is a facilitated type of diffusion, when we require some molecules, some proteins for the transportation, at that time the response and stimuli is very important. So, in this plasma membrane is also a very important function about the respond and receive of the stimuli. The properties, there is the hydrophobic and hydrophilic property, which is a major component of the plasma membrane. Because of that component, some molecules, they can't easily diffuse into the plasma membrane, because hydrophobic nature of the tail molecules which are present inside the plasma membrane, they doesn't allow the hydrophilic molecules to penetrate inside very easily. So, for such molecules, a carrier or a channel is required, which we are going to learn in our next slides. So, here the hydrophobic and hydrophilic head and tails are present, which are layered by, which are also called as the phospholipid bilayer. Bilayer means they are having two layers, one which is hydrophilic, that is the water loving, and one it is the hydrophobic, which is the water hating molecules. It is also a selective permeable. Now, one thing I want to tell you, the difference between a semi-permeable and a selective permeable. Semi-permeable means only some molecules they are allowed to transport from in to out and out to in. Okay, that is a semi-permeable. But selective permeable, it is the permeability of transportation of only such molecules which the carrier is present. If you want to transport a glucose in the cell, there should be a glucose transporter present in the cell membrane. Then only it can transport the glucose molecule. So, that is the type of selective permeability. Means only the carrier or your channel, if it is present to carry the molecule, only such molecule can be transported in this selective permeability. Structure, the fluid mosaic model, lipid bilayer, which contains the phospholipid is of 75%, the cholesterol it compromises 20% of your plasma membrane, and the glycolipids and the glycoproteins, they are having only 5% in your plasma membrane. Now, the fluid mosaic model, which was very earlier and which is the standard model for your plasma membrane, which we are going to see. The existence of plasma membrane was identified in 1890s and its chemical components were identified in 1915. The principal components identified were lipids and the proteins. The fluid mosaic model of the plasma membrane has the phospholipids, cholesterols, the proteins, the lipids, the carbohydrates that give a membrane a fluid character. Because of these structures, they are giving a fluid character. It is called as the fluid mosaic model. Plasma membrane have the thickness of 5 to 10 nanometer in thickness. The scientist Singer and Nelson, they explain the fluid mosaic model. Now you can see in the diagram that there is the various components present in the membrane. You can see a glycoprotein, a globular protein, a carbohydrate, a glycolipid, a cholesterol, phospholipid bilayer which comprises of hydrophilic head and hydrophobic tail. Then you can see a surface protein, a integral protein which is embedded inside the bilayer, and the peripheral protein which is outside the plasma membrane or the phospholipid bilayer. Then a protein channel which is acting as a transporter, the alpha helix protein, the phospholipid molecule. Okay, all these, they are the components of the plasma membrane. Now you can see in the diagram, it is the fluid mosaic model of the plasma membrane which can tell you the various combination of the fluid. How the various components, that is the phospholipid, all the cholesterol, all the proteins, they are present in the particular layer and which gives a fluid combination in that layer. So due to which you can call it as the fluid mosaic model. Now you can see the layer, the lipid bilayer which is called as the phospholipid bilayer, which in that the two layers they are attached, in which all the hydrophobic layer they are inside, which is the tail, and the head comprises the hydrophilic and the tail which comprises the hydrophobic end. Now you can see a glycolipid and a glycoprotein present on the head of the plasma membrane, which is the protein with carbohydrate attached, and glycolipid is lipid with carbohydrate attached. Then there is the peripheral and the integral protein, which is called as the peripheral membrane protein and the integral membrane protein. Peripheral which is on the surface, and integral which is embedded or which is inside the plasma membrane. Then you can see a protein channel. Protein channel, it is used when there is a facilitated type of diffusion. Facilitated means we are using some molecule we are facilitating a type of diffusion mechanism which is the in which the various molecules they are used for the transportation of the substances. Okay, this is not a simple type of diffusion. It require a carrier to diffuse the molecules. So for that we are using or bacterial cell using a protein channel. So this is the plasma membrane structure which comprises all the things in the diagram. So now we are going to see some points about the plasma membrane. Plasma membrane is highly selective filter process that permits the nutrients and remove the waste products from the cell. It maintains the hemostasis. Hemostasis means the equilibrium or the stable condition of the cell. Hemostasis, when it is acquired, at that time your cell would be very stable. Okay, it plays an important role in cell-to-cell communication. It detects the chemical messages arriving to the cell surfaces. Now the principles of membrane transport. Now we are moving towards the transport mechanism. We have seen the basic structure of the plasma membrane. We have seen the components of the plasma membrane. We have seen the functions of the plasma membrane. Now the main function, that is the membrane transport or the various transport mechanism, we are going to learn in our today's session. The two factors determine whether a molecule will cross a membrane or not. The two factors, one, it is the permeability of the molecule in the lipid bilayer, means which the hydrophilic and phobic. If a molecule can penetrate through the hydrophilic and phobic layer, then the molecule can go inside. Okay, so the permeability or the acceptance by these molecules from these molecules which are present on the plasma membrane, they give a signal and then the molecules they are penetrate inside the cell. The availability of energy source. Now if we are talking about active diffusion, it require a energy, it require a ATP. So there should be an energy source, then and then only the membrane transport can be done. Now we are going to talk about the membrane proteins. Proteins determines the membrane specific function. So cell membrane and cell organelle have the unique collection. So we have seen already the peripheral and the integral protein. Peripheral, they are loosely bound on the cell membrane, okay, or they are attached to the cell surface, which are also called as antigen. Any antigen or any causative agent or any pathogen when enters in your body, on the basis of the peripheral protein, it is detected by your body that it is the antigen or it is the foreign material which is penetrated inside your body. So, the peripheral protein is seen on every cell, and that is the identifying quality of the any organism or any eukaryotic cell, that is the peripheral protein. Integral protein, that is the penetrate lipid bilayer, and it is usually cross the whole membrane. They are the transmembrane protein and they transport protein, that is the channels and the permeables, that the porins, pumps, all they are the integral protein. Now we are going to see about the types of transport. Now when we are talking about the types of transport, there are two types of transport. One, it is the passive, and one, it is the active. If we are talking about the passive transport, the simple diffusion, the facilitated diffusion, the osmosis, the dialysis, the filtration all come under the passive transport. Because passive transport mechanism, it doesn't require any type of energy. It is the very easy process, but it is a slow type of process. Okay. In active transport, there is the primary active transport, secondary active transport, and the endo and exocytosis. Okay, in endo and exocytosis, we can learn about the phagocytosis, the pinocytosis, all that. So today only we are going to cover about the passive transport. In passive transport, we are going to learn only about the simple and the facilitated type of diffusion mechanism. Function of the membrane protein, they can peripheral and or integral, ion channels, all this we are going to learn in our transport mechanism. That about the proteins we have seen, then the ion channels which allow the flow of only the charged molecules. Okay, the charged molecules can flow in and out by using the ion channels. Carriers which can only move the polar and the large molecules. The molecules which are having the polarity, that is the hydrophilic nature. Example, glucose, nucleotides, they are the polar, which are having the large molecular size. So, they are going to be transported by using a carrier. Selectively, more polar substances cross the membrane by using a transporter. Now, we are going to see the functions of membrane protein. A receptor specific to various molecules, that is the ligand substances that binds to a receptor. Linker that helps the anchor cells together by bonding the proteins or the filament together. Cell, that is the identicary markers, majorly that is the glycoprotein and the glycoripid. They recognize the foreign cells and formation of the tissues. It is done by the cell. This is the diagrammatic way how the functions of the membrane protein. The membrane protein, first it is doing the transportation, that is the transport of nutrients and the waste from outside, inside to out of the cell, and the nutrients from outside to inside the cell. The transport mechanism, it is done by the two ways, active and the passive, which we are going to learn today. Then the next function is the enzymatic activity, signal transduction, cells to cell recognition, intercellular joining, and the attachment of the cytoskeleton and the extracellular matrix. But in your topic, in our subject, we are going to only learn about the transport mechanism, that is the metabolism. In this subject, metabolism means the collaboration of the catabolism and the anabolism, which are adding something and subtracting something. Okay, in in summation, it give a term, that is the metabolism. So, in metabolism only you are having the function, that is the transport of the nutrients from within the cell. So, only we are going to learn the main function, that is the transport mechanism. Now, we are going to learn about the membrane permeability. Selective permeability means only some substances can pass. Permeable, which substance they are easily permeable? They are the non-polar uncharged molecules, that is the oxygen, carbon dioxide, steroids. Non-permeable material, that is the large polar ions and molecules. Slightly permeable, small uncharged polar molecules, that is the water, urea. Membrane transport, we are already discussed that there are two types of transport mechanism. One, it is the passive, and second one, it is the active type of mechanism. In passive, it doesn't require any type of energy. Substances move down the concentration, that is they are moving along with the concentration gradient. Means they are moving from the high concentration gradient to the low concentration gradient. Okay, that is the Brownian motion, in which the substances, they are moving along the concentration gradient, within the concentration gradient. In active transport, it is exact opposite, because here the molecules, they are moving against the concentration gradient as they are moving from low to high. In passive, they are moving high to low, and in active, they are moving from low to high. Here the energy is required for the substances to cross the molecule, because they are moving. Example, I am giving you the example of sponge. If a sponge is put into the bowl which is filled of water, the sponge will automatically absorb the water molecule, which is a type of a passive diffusion or the passive type of transport, in which automatically the sponge is taking in the water molecule or water from the bowl. So here it doesn't require any type of energy. Okay. So this is a type of passive diffusion. Now if we want to dry the sponge, we have to squeeze it. So for squeezing, we require some type of energy. So it is a type of active transport mechanism. Okay, here we are investing energy, here we require an energy, and then only we can remove the water from the sponge. So in the same way, active transport also require energy, and it moves against the concentration gradient. Example is endo and exocytosis, and here we use of various vesicles to transport the substances from in and from out of the cell. You can see in the diagram, there is the passive and active transport. In passive, you can see the two types: diffusion through the lipid bilayer by a simple diffusion mechanism, and one, it is the facilitated, in which we are using a carrier or a transporter. Okay. In active transport, you can see molecules, they are moving from low concentration to the high concentration, and the movement is directly dependent on the ATP. If there is presence of ATP, that is the adenosine triphosphate, that is the energy, at that time only there would be the movement of the molecules from low to high. Okay. And in passive, you can see the movements of the molecules from high to low. In simple, there is requirement of no carrier or the channel, because they are the gaseous molecule, they are the non-polar molecules, and last but not least, they are the polar with small size molecule. But in a facilitated type of diffusion, the two types of molecules, they would be transported. One, it is polar and large molecule, and second one, it is the charged molecules. Now the passive transport, method of passive transport, we have seen the four method: diffusion, osmosis, filtration and dialysis. Today we are going to cover about only the diffusion type of mechanism, in which simple and facilitated type of diffusion. In the simple, it is the lipid bilayer, there are the protein channels present. The protein channels, they are maybe leaky and they may be gated. Leaky means they are open, always open. Okay. Gated means there is gate. For example, a house, if there is a gate, when a person enters, at that time only a gate would be open or closed. But if there is no gate, the person can directly go inside and come out. Okay. So here also the same, the leaky and the gated channels. The gated channels, again, they are classified in the two types. There are three types, but one, that is physical, or the physical gated channel, it is not generally termed. Here two gated channels, they are mentioned, that is the voltage and the ligand. Voltage means when there would be a potential difference in the cell. The potential difference, if it is there outside the cell, then the voltage gated channel would be open, because there is a stimuli or the response to that channel or to that proteins. Okay. And ligand gated means there would be it is a substrate specific. If the substrate is present, then it would be open, and the transport mechanism would be going on. Now there are the two types of transport mechanism. One, it is mediated, and one, it is non-mediated. Non-mediated, that is the oxygen and all the molecules, they it is a chemical potential gradient through they can cross the cannot occur against a concentration gradient. Mediated, that is the high to low passive and the active. Passive mediated transports substances that are too large or too polar to diffuse across the bilayer. They are transported by the proteins or the carriers or the permeases or the channels or the transporter. That is the facilitated type of diffusion, in which your ionophores, porins, ion channels, aquaporins and the transport proteins, they are used. For facilitated type of diffusion, all these molecules or all these substances, they are required for the transportation mechanisms, because they are large, they are hydrophilic, they are polar and they are charged. Passive transport, it is the fundamental movement of ions and other molecules, substances within the cell along the concentration gradient without any external energy. It is also known as passive transport. In passive transport, movements of molecules, they are across the semi-permeable, down a concentration gradient, means along the concentration gradient from an area of higher to lower, and here no energy is required. Now we are going to talk about the diffusion. In diffusion, we are going to talk about the types of diffusion. In that, we are going to talk about the simple and the facilitated type of diffusion. In simple diffusion, substances, they are moved freely across the cell membrane. That is the non-polar, the hydrophobic, the small uncharged molecules. That is oxygen, carbon dioxide, gases, steroids, fat-soluble, vitamins, water, urea, small alcohols and etc. In facilitated, using a membrane protein to a H2 right across the membrane. Example, polar and the highly charged molecule. Now we are going to focus on the simple diffusion. Diffusion, that is the movement of substance down a concentration gradient. It is the steepness of the concentration gradient, temperature, mass of diffusing substance, surface area of diffusing substance, diffusion distance, etc. Simple diffusion is this spontaneous phenomena where a small hydrophobic molecules, they are moving from a higher concentration gradient to the lower concentration gradient. All the molecules have the motion called the Bowman's movement and a concentration gradient. Transport across the membrane without any assistance of the membrane protein. Here no membrane protein would be required for the transportation of such molecules. No external energy is required. Diffusion stops when there is equilibrium is achieved. Means when the stability of the cell or when the molecules, they are same outside and inside, that is the equilibrium state, when it is achieved, the diffusion would be stopped. Factors that affect the diffusion rate includes the temperature, the concentration, the molecular size and the diffusion distance. Simple diffusion, you can see in the diagram that the movement of particles from high to low concentration without a protein there is doing. The concentration of the molecules, that is higher outside the cell, and the inside there is a requirement of such nutrients or the proteins. Now from the outside to inside the hydrophobic tail, they are giving permeability for such molecules to penetrate inside the cell, and the molecules, they are penetrating from outside to inside till you can see the third diagram, in which the equilibrium condition is attained, and at such condition the diffusion would be stopped. Simple diffusion is a passive type of diffusion. Example, gases can penetrate, that is carbon dioxide or the oxygen or the N2. Non-polar molecules such as steroid hormones, example testosterone, progesterone, aldosterone, all these steroidal hormones can penetrate easily through the cell membrane, plasma membrane. Polar but small molecules, which includes the water, urea, ethanol, they also can be penetrate easily. Simple diffusion, that is directly proportional to the lipid solubility and that is the membrane permeability, the magnitude of the driving force and the membrane surface area. It is inversely proportional to the membrane thickness and the size and shape of the diffusible molecule. Means it is directly proportional to the membrane permeability. Membrane, how much permission or how much permeability is given by the membrane, it is directly dependent, it is inversely dependent upon the membrane thickness, how thickened the membrane is and what is the shape and size of your molecule which going to be diffused inside the cell membrane. Now the next, the one half of your diffusion we have end up. Now the next one, it is the facilitated type of diffusion. Facilitated means you are require having some protein to facilitate a diffusion mechanism. To transport some molecules, we require some carrier or some molecules which can facilitate your diffusion, which can speed up of your diffusion. Okay. Now they only allow the large hydrophilic molecules to cross the cell membrane. Molecules bind with a specific protein carrier at the cell membrane. The combined molecule, now they are fat soluble and they can diffuse the other side. Movement is from higher concentrated to lower concentration. No external energy is required, and diffusion rate depends largely on the number of protein carriers available. When all the protein carriers are bound, saturation occurs and the diffusion rate stabilizes. In facilitated diffusion, the polar and large molecules, that which includes the nucleotides, which includes the glucose, all the carbohydrates, they are diffused by using a transporter or a carrier. And the charged molecule, example sodium, potassium, calcium, chloride, chlorine, all these molecules, they require some ion channel to facilitate the diffusion. Okay, but here the no energy is required. So we are going to learn about these two types over in facilitated diffusion. Types of facilitated diffusion: polar and large charged ions, which are I have already told you. For these two types, we are going to use a carrier and a channel. A carrier, it moves down the concentration gradient on a carrier protein. Example, your glucose, fructose and some vitamins, they are transported inside the cell by using a carrier or a transporter. You can see in the diagram that a carrier protein, it is allowing a glucose molecule to bind and to go inside the cell. Now what is happening over here? That there are some transporter. Example, for glucose, GLUT transporter is going to use. When a GLUT transporter is present, it can only penetrate the glucose inside the cell. No other molecule by using a glucose transporter can transport the other molecule. So it is a selective type of permeability. Now here, when the GLUT transporter, when a glucose molecule is attached to a GLUT transporter, it is very specific. The transporter, it is a very substrate specific and the stereo specific. Substrate specific means that transporter can only take up a specific substrate, and stereo transporter means form of your substrate. If it would be L form or if it would be D form. If the glucose is L form, the L form of transporter can be only used. So here, if your glucose molecule is attached to your transporter, then the conformation would be changed, and the conformation change like this, that the glucose molecules is penetrated inside the cell. And again, when it is empty, again the conformation change, and again it goes and takes up the glucose from outside the cell. So in such way, the transport mechanism is on and off on the basis of the substrate availability outside the cell. Carrier proteins, carrier or they are also called as the transporter proteins. They move the uncharged molecule, example glucose protein, change the shape and allow the molecules for transport. It acts like a gate or gate of the door. It also called as the permease or the transporters, because it is allowing multiple spanning membrane protein. It can bind to a multiple membrane protein. It is very substrate specific and stereo specific, which already I have told you. Carrier proteins, it transport protein alternate between the two conformation. Erythrocyte glucose transporter, example GLUT1, it is present on the RBCs, and what they are doing, it is transporting the erythrocyte from the to the RBC. Has the glucose binding site on both the sides of the membrane, okay, and it is a tetramer in form. You can see in the diagram that a glucose is attaching, and then there is the conformation change of the GLUT transporter, and when the GLUT transporter is empty, again the conformation change, and again it would be bind again to another glucose molecule. Carrier protein, it is a type of facilitated transporter. Here also you can see a carrier protein. It is taking up a substrate inside it, and when the substrate goes inside, the conformation changes and the substrate, it is penetrated inside the cell. Again the conformation change, and again it pick up another substrate. Now, if I am taking the example of glucose transporter, there are 5 transporters present of the glucose, that is called as GLUT1, GLUT2, GLUT3, GLUT4, GLUT5. In GLUT1, the location we can see over here. GLUT1, it is located on the RBCs. It is doing the erythrocyte transportation to the RBC. GLUT2, it is present on the liver cell. GLUT3, it is in the brain, which are doing the neurons. in the neurons, the axons, you all of you know the cell body contains the GLUT transporters. GLUT5 is present in the muscles, skeleton and cardia, which is trying to keep your insulin level at the equilibrium, stable conditions. Okay, GLUT5, that is intestine, kidney and testes. GLUT5 has the special function. Okay, what function? That it is transporting the ribose sugar, fructose sugar. GLUT5 is transporting the fructose sugar. It can't transport the glucose sugar as it is mentioned at GLUT5, but it is transporting the fructose sugar towards the kidney. Okay, so this is the special function of the GLUT5. Channel, now we have seen about the carrier, that is the transporter. Now we are going to see about the channel mediated. In the channel mediated, it moves again, it is the type of passive diffusion, so the concentration it would be from high to low. Okay, molecule move small charge molecule, example ions. Here from the channel only the charged molecules they are going to transport. So charged particles need help in crossing over the hydrophobic area, so channel is required. Particles transported have the opposite charge to that of the protein. It acts like a tunnel. You can see in the diagram, again a protein channel it is used, and the opposite charge which is present on the substrate against the protein channel, it will be attracted towards it, and that molecule it would change the conformation and again it would be penetrate inside the cell. Okay, so this is the type. Cytoplasm means inside the cell. Okay, that is and the extracellular fluid means outside the cell. You can see in the diagram how the molecules they are penetrate inside the cell. You can see the charge, only the charged molecules they are going to penetrate by using a channel protein. Now in channel protein, one example we are going to see about the ion channels which are gated. Now when we are talking about the channel protein then again they are of two types, one it is gated and one it is non-gated. When we are talking about the gated one means which open and close. So in that we are having the three types. One it is the ligand, second one it is the your voltage, and third one it is the mechanical gated channel. And in non-gated there is no types. Directly the molecule can penetrate inside and outside by the channels, and there is no opening and closing mechanism here. So in gated, that is which are having a opening and close mechanism, they are mechanosensitive channels which open in response to membrane, that is touch, sound, osmotic pressure, example your skin, your ear. Okay, when there is physical or mechanical touch or mechanical sound, at that time these channels opens and the molecules they are transported inside. Ligand gated channels, they have in response to extracellular chemical stimulus, they are present on the neurons. And in your neuron, they are present on the cell body. Okay, on the cell body, these types of channels or these types of ion channels they are present, and they are doing the neurotransmission. And the last one is the voltage. Signal gated channel open, for example intracellular binding of calcium, and the voltage gated channel open in response to membrane potential change, transmission to the nerve impulses. They are also present in your neuron, and on the neuron they are present on the axon. On the axon of the neuron, your voltage gated ion channel are present, which are transporting the various ions from outside to inside your body. Now you can see in the diagram ion channels, for example sodium ion channel, potassium ion channel. It is very specific. Sodium ion channel can penetrate only your sodium molecules. Potassium ion channels can penetrate only your potassium molecules. Okay. Now we have already learned about the all passive transport. In passive transport we have seen about the simple diffusion, and the second one it is we have seen about the facilitated diffusion. In facilitated diffusion we have seen two types. One it is the carrier or the transporter, and second one it is the channel, that is the ion channels. Okay, differentiate between the mediated and non-mediated. Speed and specificity, structurally similar but take up glucose in much faster, that is the mediated. Okay, now in this graph you can see the two type of graph. One it is the straight line and one it is the sigmoidal curve. When we are, if I ask you one question that which type of graph represent a simple diffusion and which type of graph represent a facilitated type of diffusion, For this, I will be giving you the answer that if your X line indicates the concentration of your substrate and if your Y line indicates your transport rate. Okay. Now, one thing I want to tell you that in simple diffusion, the transport rate is very slow, that is 10 raised to 0 to 10 raised to 2 molecules per second. They are transported. And in passive transport, the rate it is 10 raised to 6 to 10 raised to 7 molecules per second. Okay, so facilitated type of diffusion is fast because it is carried by a carrier or a channel, and this one is a natural process. It increases as the concentration increases. Now in this graph, this is very important for you, this point, because you can detect the simple and facilitated type of diffusion by using this mechanism. Now if I am talking about the X-ray, which is giving the concentration of the substrate, if I am using the substrate concentration, automatically upon my substrate increasing substrate concentration, your graph would be increased, and it requires some time period. Means the straight line graph indicates your simple type of diffusion, and the sigmoidal curve, because sigmoidal curve is facilitated type of diffusion, the Vmax is already attained. Now you can see in the graph the Vmax is attained, and as soon as the substrate concentration is given, directly the Vmax is attained because it is substrate specific, it have a carrier molecule, and it is very fast, that is 10 raised to 7 to 10 raised to 6 to 10 raised to 7 molecules per second it is going to transport. So here Vmax is attained, so it is a facilitated type of diffusion. In your simple, Vmax can't be achieved because we are increasing the substrate molecule, and according to our substrate concentration the diffusion would be take place. And but in your facilitated type of diffusion, it is substrate and stereo specific, so given substrate would be transported within a second. So it will attain a Vmax and it will be stop over there. Okay, because the equilibrium it is attained. Now which factors can affect the rate of diffusion? The molecular, the membrane related, and the gradients. These are the three main things which can affect your rate of diffusion. In molecular, the lipid solubility, the molecular size and the weight, and the temperature. These are the three things on the molecular basis can affect your diffusion. Thickness of the membrane, surface area, and the concentration gradient can affect your membrane related. Concentration gradient, pressure gradient, and the electrical gradient can affect your diffusion rate by the gradient methods. Now we are going to see the last part of our today's session, that is the direction of the transport. The direction of the transport can be of three types: uni, sim, and anti. Uni means only a single molecule is going to transport in only one direction. Sim port means two molecules they are going to transport in the cell but in same direction. And anti means again two molecules they are going to transport inside the cell but in the opposite direction. Okay, example of uni is all channel proteins, example of sim is sodium and glucose importer, and example of anti is sodium and potassium pump. You can see in the diagram, that is the sim porter, the anti porter, and the uni porter. Now again I can show you the diagram. First diagram is of the uniport. The molecules they are transported in the same direction, only one molecule. In sim porter, two molecules they are transporting but in the same direction again. And in the antiporter, two molecules they are transporting but they are transporting in the opposite direction. That is also called as the co-transport. Now we are going to see about the distinguishing features of the active and passive transport. In passive transport, no expenditure of energy molecules are there. It takes place long and concentrated and electrical and pressure gradient. It is having a carrier. It may require a carrier. The rate of proportional, it is dependent on the concentration difference. In active transport, the expenditure of energy is required. Can takes place against the concentration gradient. Passive, it is along the concentration gradient. Here compulsory a carrier is required. The rate of proportional of the availability of a carrier and energy, it is the Vmax. So these were the distinguishing features of the active and passive. Now we are going to see the distinguishing features of simple and facilitated type of diffusion. Simple type of diffusion is a passive transport. It only transport the small molecules. No carrier is required. The rate of transport is directly proportional to the concentration gradient. Example is lipid soluble, that is the gases. The lipid insoluble, that is the urea, which are polar but small molecules. In facilitated type of diffusion, passive transport, it is also a passive transport mechanism. Here large molecules they are transported. Here carrier is compulsory required. Initially the rate of it is proportional to the concentration gradient till it achieve to the Vmax, that is the saturation of the carriers. Example is glucose and amino acids. Now you can see in the a diagrammatic way of both type of difference mechanism. One it is a simple and one it is the facilitated. Simple diffusion without the help of the proteins and facilitated diffusion with the help of carrier or channels. Okay, you can see a channel and a carrier in the diagram, which is penetrating the molecules inside the cell, but both of them they are penetrating by only one mechanism, that is the higher concentration to the lower concentration and along with the concentration gradient. And third similarity is without any energy, no need of ATP or no need of energy. These are the three things which is similar between the simple and facilitated type of diffusion. For our today's session, I am giving you some assignment. You have to describe the structure and the function of the plasma membrane. You have to describe about the fluid mosaic model. You have to explain how the hydrophobic interaction determine the membrane structure and the transport mechanism. You have to describe how proteins are arranged in the membranes and how they contribute to the membrane functioning. And you have to write in detail about the passive type of mechanism, passive transport mechanism. So here we have taken all the methods of transport. Now we are going to see what we have learned in our today's session. We have seen about fluid mosaic model. We have seen its characters, its functions. Okay, we have seen membrane transport mechanism. We have seen the active and passive. In that we have learned about a detailed mechanism of passive transport, in which we have seen about the simple, the facilitated. We have seen the direction of the transport. We have seen its distinguishing characters of active and passive, and the simple and the facilitated type of diffusion. We have also seen the similarities, the same things about the simple and the facilitated type of diffusion. I think all of you have now know about the diffusion, that is the passive transport mechanism of the cell. I think I have cleared this topic up to you. Now, if you have any questions, you can ask me about the passive transport. So in our tomorrow's session, we are going to learn about the active transport mechanism, which is requirement of a carrier and also a compulsion of ATP. In the active transport, compulsion there is the carrier molecule and the ATP molecule, which we are going to see in our tomorrow's lecture, in which we are going to see the pumps, the endo and exocytosis, the phagocytosis, the pinocytosis. Okay, all things which we are going to see. We are going to see the type of active transport, which includes the primary, the secondary and the electrochemical gradient. We are going to also see about the ATP driven pumps, which includes the P type, the F type and the ABC transporters. Okay, so here our today's session is over. Again we are going to continue about the active transport in our next session. Thank you for attending the lecture.